Hey guys, welcome to the video on Chapter 8, Lesson number 3. So, for this lesson today, guys, I'm going to be going over some information about experimental design and how to create your own experiment. But before I do that, let's go ahead and go over the opening questions. So, you want to go ahead and pause the video now, and you want to take a crack at these questions. Okay, so let's go over the answers to the questions. The first one, what is energy? Energy is the ability to do work, such as moving things around and stuff like that. It's a very, very simple idea, but it's something that you kind of have to think about, okay, what actually is energy? What are these things we keep on talking about energy? And energy is just the ability to do work. The next one is, what is the name of the molecule that your body uses for energy? Well, the answer to that one is ATP. If you said adenosine triphosphate, you are also correct. It's the exact same thing. ATP is adenosine triphosphate. And the last one is, what are the two types of metabolism? You have two types. You have catabolism, which is breaking down molecules, so taking big molecules and making them small. And you have anabolism, which is building up molecules and making them big. Okay, so I have three goals for you guys today. The first one is I want you guys to be able to create an experiment to test a hypothesis. Now you're probably thinking that you've already done that before, and you have, which is good. But this time around, you're actually going to be running this experiment. So it's going to be different this time around. You're going to be going to the lab, you're going to be doing an experiment, but you are going to be creating the experiment and running it. I'm not going to be giving you a sheet of paper with directions on it to tell you how to do it. You are going to be creating your own experiment based upon what you know. With that being said, that makes it a little bit harder than a lot of our labs have in the past. So I'm going to be doing things a little bit differently here. The last goal I have for you guys is you're going to be able to analyze a self-created experiment. So not only are you going to create and run an experiment, you are also going to do the math to tell you what happened in this experiment. Now this all sounds like it might be challenging, but you've used these skills before. This is just going to be the very first time that you're going to actually be putting them into practice. Okay, so a quick review, guys. Hypothesis. A hypothesis is just an educated guess of what might happen. So it's you guessing about what might happen if you do something. An experiment is a test to see if a hypothesis is correct or incorrect. An independent variable is a part of the experiment that you are going to change to change. So in the experiment we did on enzymes, this would be a good example would be the amount of acid or concentration. We changed those variables. The dependent variable is what you want to see change. So in the previous example with our enzymes and acids and bases, the acids and bases were our independent variables. We were changing that, and the dependent variable was how fast the reaction was taking place. Okay, so how did you create your experiment? Well, the very first step, guys, is you want to find out what you're supposed to do. That sounds kind of common sense, but you sit there and think about it. You actually sit down and think, okay, what do I have to do here? And this is called your research goal or your question. So, for example, if you are asked to find out how fast an enzyme works, you will need to find out how you could go about measuring this. And a good place to start is look at the chemical formula. And you'll find this advice right here will help you a lot for this chapter. Because in this experiment, you will probably figure out how to do it when you look at the chemical formula. The second step is you want to actually create a hypothesis. And for this situation, I'm going to give you a formulaic hypothesis you can create. Something you can just put in words and usually it'll work okay for you. And so you want to make it like this. So when I do blank, this blank will occur. So when you sit there and say, when I do independent variable, this dependent variable will occur. So in our example with the enzymes, and you can see when I increase concentration, this lowering of concentration rate will occur. Period and simple. Okay, the next step. You want to design an experiment to see if your hypothesis is right. So the first thing you have to do is you have to make sure you know what you're changing. Know what is different from one part of the experiment to the other part of the experiment. And this right here is your independent variable. The second thing you want to do is you want to know what do you think is going to happen? What's going to happen when you change something? This is going to be your dependent variable. So you want to make sure you know what these two are beforehand. And if you followed my formula, it's pretty simple. This will be your independent variable, and this will be your dependent variable. The next thing you want to do is you want to create your own procedure. Now you've seen a procedure before. Whenever I give you guys a lab, it's the part of the lab that tells you basically what you are supposed to do. Now this time around, guys, rather than me giving you a procedure, you're going to have to create your own. So you want to write down what you're going to do and in what order. That way when you get to the laboratory, you kind of read it off like a cookbook. You just read over, say, okay, next I'll do this, next I'll do this, next I'll do this. And the next thing, the part of that is you have to decide what tools you're going to use. What scientific instruments are you going to use? Now don't worry, I'll have actually a list of supplies up here that I think you're going to need to have for this lab. So this part shouldn't be too difficult, but it could be tricky if you don't quite know what you're doing with the parts up here. The third thing, I guess experiment part 3 up here, is you have to decide how you are going to analyze your results. How are you going to find out what happened? Because you have numbers, but numbers only go so far. You have to actually take something and do something with those numbers with math. And what you can do is you can create a table of some kind, and you want to create a table that you can put all of your data onto. But then you want to create a graph of some kind. So you have to decide, what sort of table am I going to use, and what sort of graph am I going to use to interpret that table? And this is something you're going to have to find out for your experiment. For this experiment, guys, I'd recommend one of 2 types of graphs. Now, one of these is better than the other for this experiment. I'll be curious to see what you do and which one you choose. But either you want to go with a bar graph, which is the one you have the bars that go up like this, or even line graph, which is the one you have a line that goes up like this. You're going to have to decide which one of those is the best for your information. The last thing you do for your experiment is you have to write a conclusion. The conclusion is simple. This part here, you just say, what happened? So if you ran your experiment and you found out that the more enzymes you put into the hydrogen peroxide, the faster it goes. This part right here is where you actually tell me that. Plain and simple. Okay, so a review of what I talked about for this lesson. First thing, when you want to create an experiment, the first thing to do is you have to figure out what you are measuring. What are you going to do? What is your goal? The second thing is you have to create a hypothesis. The third thing is you have to make an experiment to test your hypothesis, and make sure you know what the tools to use are. And I was showing you before, I'll give you tools up here that you can look at and decide which ones you want to use. You then want to create a procedure of what you're going to do when you get to the laboratory. Once you've done that, you're going to figure out what kind of table and what kind of graph you're going to use, and then you go do it. You go to the lab, make it happen. And once you have your results, you draw a conclusion based upon those results, and that's really it. So here is what your guys' activity is, and this will probably take you the next couple of days. So don't be surprised if it takes you 2 or 3 days to get this done. Okay, so here's your research goal. I've already given you the first part up here. You want to determine if germinating and non-germinating peas undergo photosynthesis. Now the word germinating means it's basically a pea that's starting to grow. It's a pea that already has little seeds growing out. It has little branches and leaves growing out of it. And non-germinating is basically means that it hasn't actually started growing yet. So which one of these do you think is going to undergo photosynthesis, or are both? You have to find out which one is going to. So what you have to do is this. You have to create a hypothesis based upon this research goal. You have to create your own experiment with a procedure that explains what tools you're going to do and how. You're going to figure out what tables you're going to use. You're going to figure out how you're going to graph it, and then you're going to run the experiment. Now, I'm going to make this part of your guys' assessment mark, so it's going to come as part of your quiz mark for this chapter. So you're going to turn in what you've done to me either on Edmodo or on a piece of paper, depending on how you chose to do it, because this is a really important thing, guys. This is science. You've probably thought that science is just memorizing information, but at the end of the day, science is a process. Science is actually doing something. And this is the first time, guys, that you guys are actually going to be doing science as scientists. I've given you a question, and now you have to find the answer to that question using the tools I'm giving you. So this will probably take you a couple of days, so don't be surprised if it's a little bit challenging. And as always, please, 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 please ask me for help if you need it. I'm here to help, and you are more than welcome to work in groups, because I suspect you guys should work in at least groups of 2. I would like to have at least 3 though per group. So if you get to this lesson before somebody else does, or before the person you want to work with does, you can go ahead and move on to the next lesson, and when they get here, you can go ahead and pair up with them, and you get the work done in the lab. All right, I'm done talking. Thanks for your attention. Have an awesome day.